Welcome to the front row. I'm Jamie Williamson. I'm the chair of structural biology here at Scripps, and I'm your host for today. So it's got a good group. It looks like about 200 and odd people here in person. We had a few thousand that have registered to join us online, so hello to everybody in the virtual audience. So today we're going to talk about proteins in motion, and Dorothee Kern is an expert, and she's moved to Scripps here about a year ago. And so I was trying to think of a good example of what's going on. And so some of you must have heard of this, but in 1878, there was a photographer, Edward Muybridge, and there was a debate among the cognoscenti in the horse racing field about whether the horse always had one foot on the ground or not. And so he set up these tripwire cameras, and I'm sure you've seen these pictures of the horse, and there's actually a point where all four hooves are off the ground and in the galloping motion. And so you can imagine if you just have the picture of the horse, or just in your mind's eye, the picture of what the horse looks like, you can't actually figure out how it's running. And it took this high-speed photography in order to resolve this issue so you can actually see what's happening. And Doro uses nuclear magnetic resonance to essentially look at the movies of the protein in motion. Let me give you another example. Imagine you saw a picture of me and I was like this. Then you'd say, wow, Professor Williamson, he's got some dance moves. But to really see it, you have to see me actually dance. We're not going to do that. So the point is, every front row that you come to, someone will show a picture and there's a protein. And it's a paradigm in structural biology that there's a relationship between structure and function. If you know the structure, then you learn something about the function. Everybody shows these freeze-frame photos. And so Doro's lecture is going to be all about how you can exploit the intrinsic motions of the proteins to understand yet even more about their function and to do some amazing things with drug discovery. Now, Doro is an athlete of some repute. I think she's going to tell you about this. I've been a friend of Doro's for about 30 years. And that's despite the fact that on several occasions she's tried to kill me. So she's, in addition to being a good athlete, she's a coach. And so we were skiing at an NMR conference at Keystone, and she's taking me down these runs, and she goes, okay, follow me. And now all of a sudden we're going down chutes and trees and rocks, and I was just fearing for my life. And she goes, you did fine, you did fine. But she's a great coach, and I think she'll give us a little bit of highlights, maybe, of some of your exploits. Okay, at the end, I'm going to come back, and then Doro and I will be here to have a little discussion and take questions from the audience. So stick your hand up. Someone has a mic, and they'll come and get the mic to you. So without further ado, please welcome Doro.
I'm very excited to see the room full of people who are excited to learn how studying the dancing proteins will help us all together to happily and healthy dance through life. What is a protein? You probably know protein as a really good source of nutrition, your steak, your cheese, or of course the key ingredients for muscles demonstrated here by my older daughter. So yes, I'm not going to use horses. I'm going to use my little horses, my two daughters, to demonstrate. Nadia, as an alumnus at UC San Diego. But what are structures? I will tell you that proteins do much more. Proteins basically control everything that is going on in your body, okay? And they're made out of 20 amino acids. Here's kind of the code, right? The language of a protein. And then they fold up in this, what we always see, these static structures. But what I will tell you today is if we would sit here, we would be all dead. The entire biology happens because our proteins dance. Now, how did I get into that I want to add the fourth dimension to structural biology to make real-time movies of the atomic resolution of these proteins. It started playing me basketball when I was an undergrad and in high school and as a PhD student in the national team. So I was actually probably doing more sports and studying science, but I realized that these static structures don't give us whether I will make that basket or not. And so I got excited to really having that vision since I was an undergrad to make real-time movies of proteins at atomic resolution. And I will tell you how this geeky biophysics, which started from sports into biophysics, led me to found 2 biotech companies, Relay Therapeutics and Myanmar Therapeutics, where we actually cured the first cancer patients in the last 4 years. So the question is, why a protein dance? Here's my other daughter, our younger daughter, just a couple of weeks ago at the Olympics, and you can see, that snapshot, my other horse, but you don't see how well she is actually running. And for that, you have to turn on the TV.
Really grown as a cross-country skier in recent times,
and you will see her racing in a team sprint over the mountain.
Team sprint silver then at the last World Championships in Trondheim last year.
The mountain will become important.
Here she is with a potential podium place. A great fight back on the
So now, this is microscopically, structural biology gave us these static structures, but very similarly, we want to look at these proteins in action, and we can't just turn on the TV and ask us how to do it, right? A little fun fact, ATP is actually the currency of energy in your body, so you hopefully have a lot. And, of course, Julia consumes a lot of ATP, and the winning margin of 20 milliseconds. And this enzyme, which I will be talking about, makes that ATP. And this would actually take 7,000 years without this enzyme, and this enzyme can make it in 5 milliseconds. Now,
doing remarkably well.
The question is the dream and the journey. So when I started my PhD, the dream was to do that and the journey. And so I told you that proteins dictate basically everything in your body. So the picture when I was an undergrad was signaling is like an on-off switch. You have an inactive state, and then you have an active state. An enzyme is sitting still, membrane transport, and so on, right? So for all of these biological processes, long-term memory in your brain and so on, actually do you need proteins? But people said, okay, we understand that. But I'm challenging you. Do you understand biology by looking at these static structures? No, because here is where biology happens. And so this dream of visualizing directly how your brain functions at atomic resolution, so hopefully your long-term memory works, that you can still remember what I tell you next year. This is in membrane transporters. This is when you take antibiotics, your body has transporters which transport that back out. Signaling happens by constantly jumping back and forth. And this one is actually a protein-protein interaction in your vision, which happens in milliseconds. But then people said, yeah, for healthy function, maybe we need those dancing proteins. But for drug discovery, not really. And wait until the second half where I show you that the key to make drugs which have no side effects is actually to use the dynamic properties of a protein. Now I show you all these movies, and the question is, how do we arrive at this? We can't just take a camera and look at it. So we have to use these biophysical methods, which we are all using in my lab, to see the dancing proteins. Amy, show me some moves. I'm playing dancing in the dark because we need these biophysical methods combined with computation and AI methods to finally reconstruct these movies, okay? Now, we asked the question of why and how. Now I want to explain you a really big concept. The concept of the mountain, okay? So these static structures, which we can actually have been determining for 20, 30, 40 years by either structural biology methods, or now we can even predict it. Ooh, this one died. Okay, now I have to use this. Either with AI methods, we're sitting in the valley, but every protein has to go over the mountain. And it's trying, and it's trying, and it uses a lot of energy. And most of the time, it sits in the valley. And very rarely gets over. And the bigger the mountain, the slower the reaction. So this reaction, what I told you is 4,000 years to make your ATP, has a big mountain. And what an enzyme does, it lowers the mountain, and therefore it's easier to go over. That's the entire thing an enzyme does, and therefore these reactions are much, much faster. Now, here is this enzyme I was talking about. It's in every cell you have, and it's essential for every organism because it is essential to make ATP, okay? So it's a fully reversible reaction. I told you this amazing rate acceleration, which is still mind-blowing. So evolution evolved those proteins to do that. And when we start usually a project, we have a few structures, but a lot of things we don't know. And all the things we don't know are all the red stuff. The top of the mountain, the relative populations, how fast things are going. And for that, we are using this method, which Jamie said was the nuclear magnetic resonance spectroscopy. What can we do with that? We can measure dynamics over the entire time range from picoseconds to hours. We can measure how every single atom in your protein is dancing. And we can measure it under physiological conditions. In crystallography and cryo-EM, we freeze our samples to minus 120 degrees centigrade, so they are frozen. But in NMR, we are at normal temperatures, they are constantly active. The physics behind is identical to MRI imaging. So it's a big magnet we're using, but what we're doing in imaging, we're looking at a whole knee. I hope you don't have to be in these big things because they're pretty loud. What we are doing, exactly the same physics, but we have our little tiny proteins in this tube, put it in the magnet, and now what we are measuring is radio frequencies. Every atom has a little different tone. I'm playing you right now, you're listening to a protein, the frequency of each single atom. And this one we're converting first into a spectrum, and next, we are converting the spectrum into how fast these atoms move. And what we're doing, we're taking our little enzyme, and I say I'm in action because as soon as you give food, in this case magnesium ADP, it's doing a catalytic reaction. And I did put some data in, so because I don't want to just give you fake news, we as scientists actually buttress our stuff with real data. Sorry, Jamie. He didn't want me to put in data. I think it's important. So these are the real data. These are translated. What you can see now, statically, that the protein opens and closes. This movie here is the opening and closing of the protein. And it turns out the opening rate is identical to how fast this protein makes ATP. So what we know, the limiting step, how fast this enzyme works, is actually how it's opening and closing. And the thing which takes usually 7,000 years, this little ATP, making ATP, so I want to show you here for the chemists, we have our little ADP and it makes ATP here, whoops, it hops over. This happens in picoseconds, okay? So I leave you with these movies to have kind of a little understanding how amazing evolution happened to make these sophisticated proteins, and we are far away of designing these things. Okay. So, I was looking at these happy, healthy proteins, and here comes my husband, and, he annoyed me because he said, who cares about these dancing proteins? Where are the applications? I mean, he happens to work in pharma and biotech. So just to shut him off, I said, okay, I'm going to look at some good drugs and look at how these drugs bind to a protein, because, of course, I know the protein is dancing. And thanks to my husband, I actually came up with a new vision for drug design based on protein dynamics. I have to disclose that I'm the founder of these two companies. So that will be the first part of application. The second part will coming back to these enzymes who are that fast, which we can't really design well, but I will give you some hope that we're working right now on a therapy for everybody in the audience who's gluten intolerant, to have a little pill that you can have the good food when we go out later. Okay. Where was the problem for rational drug design? It's right shown over here in this little movie. It's called the rigid body docking problem. This drug tries to go in. This is actually an HIV reverse transcriptase. It's an approved drug against HIV. And if you look at the crystal structure, it's in the inside of the protein. You can't even see it. How can it go in without moving? It has to open up, bind, and then cover over the drug. So that's where I got the idea that protein dynamics is absolutely essential. And so I thought, let's have a look at how these proteins dance. And So the idea is basically that you have a protein which toggles between two conformations. One where the drug can bind, the little triangle of the drug, and one where it cannot bind because the pocket is closed. And I will show you that this is, of course, absolutely essential because otherwise the drug cannot bind. But the even more important and completely ignored part in all pharma industry is that second step. After the drug binds, you actually have protein dynamics in the drug-bound state. And that is actually the absolute key which I discovered. And so this led actually to those two companies. Okay. All right, so let's have a look. How did we get to that? I am not smart enough to design a very good drug. So I said, let's find a success story. And the success story is Gleevec. So Gleevec was called the multi-billion-dollar molecule because it's extremely good against leukemia. And it's one of the very few drugs which are actually selective, which only inhibit the disease one and not the healthy one. It was a big puzzle because it bound the disease one, ABL, the leukemia kinase, 3,000-fold better than the homologous kinase, SAR kinase, which is healthy and we don't want to inhibit. Now, where are all these drugs going? They actually go in the active site. And I told you all these kinases, they all bind ATP. So we have 500 kinases in our body. Think about 500, they all bind ATP. The pocket is the same. So that's why usually what happens is that you're not only inhibiting the disease one, but also the ones we need for health. This one was discovered to be selective, and the big puzzle was, if you look at the crystal structure, the binding site between those two proteins is identical. So if the binding site is identical, how can it be bind so differently? So the answer must be in dynamics, right? So that's what I thought. So we go back to our NMR experiment, where each of those peaks down here is one amino acid, and of course what we do, we take our protein, and then we titrate in our drug. And here comes a very important concept. What we found, that every signal shifted first, then disappeared and reappeared. Now what it means, that actually there are two processes. One which is fast. If I run back and forth very fast, you only see me in the middle. So a fast process on the NMR time scale, you're shifting. When I disappear here, and much later on, I come up here, that means a process is slow. So I knew right away from this 24-hour experiment, which actually an undergrad in my lab did, that we have two steps. One is fast and one is slow. Now, we just write our cartoons, and you're already familiar with that, right? And it's actually, there are actually 3 steps. But what we found out, that the fast process is the physical binding of the drug into that pocket. Okay, that's fast. That's what every drug company did with docking. The slower process is after the drug binds. Remember, I told you, you couldn't even see the drug. There is a conformational change. Part of the protein cuddles over the drug and holds it tight. And that cuddling over is the key step to make selective drugs. It happens after the drug binds. And what I show you here is now with our data that this equilibrium is 4,000-fold shifted to that right side for the cancer protein and only 6-fold shifted over here. Now comes the most important thing. The overall affinity is actually all 3 steps. This step is identical. The docking, what everybody's doing. The healthy and non-healthy docking is actually identical, 17 micromolar. Billions of dollars wasted for 20 years of making selective kinase inhibitors because every company wanted to exploit this interconversion between the pocket and non-pocket. It gives you almost nothing, right? So actually, anything we are exploiting, you make the affinity weaker because you go more and more into a conformation which can't bind the drug in the first place. In construct, if you actually dial in with a drug that you make this cuddling over, you can make a micromolar binder into a nanomolar binder by just multiplying that number with 4,000. So this is actually simple thermodynamics which we're teaching undergrads. Unfortunately, often not anymore. It's really just figuring out thermodynamics of proteins. So we figured it out. That's why this drug binds 3,000 times tighter to the cancer drug because of this slow dynamics in a drug-bound state. Now, we figured it out for a drug which is also on the market, but how can we now use this concept to make new drugs? We actually have to dial it in. And for that, we have to figure out why one protein denses differently than the other. And for that, we had to actually go back in evolution 1 billion years. And why? If you compare the cancer protein with a non-cancer protein, it has 146 amino acid differences. 70% of the protein is different. But not all of those are responsible for different dancing. We went back in evolution and calculated how these proteins looked like the common ancestor 1 billion years ago. And we made those in the lab, meaning Roman and Chris, two very talented postdocs in my lab. And then we measured the affinity of this ancestor and the other proteins to the drug and solved the crystal structures. This trick allowed us to find the 15 residues in blue here who are responsible that one protein does this and the other one does not do that. And of course, the dynamics had to be away from the binding pocket because the binding pocket is identical, so it's actually my elbow. The elbow is straight-jacketed for the one which can't bind, it can't cuddle, and able, the cancer one, removes the hydrogen bonds and therefore is flexible. So it's the flexibility away from the binding pocket which allows the drug to now be very, very high affinity. All right, since you are paid so much attention here, I'm going to give you an even better approach. I asked the question, why do we have 500 kinases? They do all the same thing. We have 500 because we're a little bit more sophisticated than a bacterial cell, and we do all these kind of different processes. So the differentiation is actually in differential regulation. And what these kinases all have, they have regulatory sites, which are away from the active site. So I said, if those regulatory sites are different, shouldn't we just target those? Because now we get much, much higher selectivity as number one. And number two, we can actually also make activators. So far, I've always told you, you're getting diseases because a protein is too active and we have to inhibit it. But there are other diseases where we actually lose the function and we need molecules which activate the protein. And that's best shown for this, on this little seesaw here, okay? So the first thing is, here is the concept, right? And the concept is, it's toggling between the inactive and active state, right? And what we need, either a little brown hair little boy who binds to the green guy and therefore shifts the equilibrium and we count that activator, or we have to find some red-haired girls calling inactivator who tips the seesaw the other way because it binds to the inactive state. So by binding to the active or inactive state, we can actually make the protein active or inactive. And so we chose this kinase here. It's called Aurora A kinase because it's essential for cell division. And it has a name because it lives on the spindles down here on the cell, the poles. And that's where it comes from, Aurora borealis. You got it? Yeah. So now we have the concept. We have our protein. It's essential, again, in cancer treatment. And now we have to screen. So how do we screen? This time we're not looking for small molecules. We're actually taking tiny little proteins. They are kind of little antibodies. And we're taking a whole collection, 10 to the power 4, there's 10,000 different variants, and we just screen which ones will bind to this protein and activate or inactivate. So which ones is the little boy and which ones is the red-haired girl. And this was a very good day. We indeed found an activator, green. It's actually a better activator than the natural activator on the cell. We are activating 15-fold. And we found 3 different inhibitors. They're inhibiting between 10 and 20-fold because they are binding to the inactive form. And then we solved the co-crystal structure, and sure enough, the inhibiting monobody shifted the entire kinase to the inactive state, and the activating monobody, the entire kinase to the active state. Now you're still following me. So we solved the first problem in cancer, which is selectivity, that we have no side effects. But there's actually an even bigger problem. Everybody is familiar with that. The really devastating things what happen with cancer patients. Chemotherapy works, and then it doesn't work anymore. And it doesn't work anymore because a protein becomes resistant to the drug because it mutates. So what is the approach to combat this drug resistance? by combining both drugs I talked to you about. The regular drug together with its active site drug, which is already giving to cancer. And why do we want to combine two? If you combine two, there is the probability that you have two mutations at the same time to render both drugs not being possible is infinitely small. This will not happen in the lifetime of a human patient. So this double drugging really, really goes after the biggest problem of the drug resistance in treatment of cancer. And then, of course, the other two really important things, one inhibits, the other one inhibits. So it's an additive effect. So we are inhibiting even better. Now, how do we do this? We're going back to our toggling back and forth. What we need to find are two drugs which work together. And I show that a little bit as this tug of war. So remember, we have the red guy, one structure, and the green, the other structure. We want to find small molecules which work together with our inhibiting monobody, meaning both of them bind to the inactive form. So like these guys, they work together. However, if we now would combine one which binds to red and one which binds to green, it would be a tug of war. And they would be actually negative cooperative. And that's what we saw. Indeed, the monobody one, which is an activator, and there's an inactivator, is negative cooperative, 16-fold. If we now use our monobody, the inhibiting monobody, together with this cancer drug, we actually have positive cooperativity. Not only that they are positive cooperative, the cell only cares about activity. Because they are both inhibiting, you're adding the effect together. And what we found, that you need 20-fold less from the chemotherapy to get the same effect. And with 20-fold less, this non-selective chemotherapy becomes selective, because now it's not binding to the other kinases anymore. So in other words, we are minimizing, again, the toxic side effects. So these were all proof of concepts experiments. And I hope I kind of conveyed a little bit to you how important academic research is. All that academic research was done in a little lab with amazing grad students, undergrads, and postdocs who actually had the passion to understand proteins. And it was not my goal to actually start a company, but because we did fundamental research, we found completely novel things nobody had considered in industry. And this novelty allowed us now to make very selective drugs, okay? So I will give you two examples. The one which is just still having me cry. So we made the first selective inhibitor against pancreatic cancer and cholangiocarcinoma. And it was about last year at Relay Therapeutics, based on protein dynamics. I'll show you in a minute. Last year, a father sent us a letter, and so he actually was terminal in pancreatic cancer, had to come off the chemotherapy he used because the inhibitor he used was not selective. So he needed to inhibit FGFR2. He got on our drug and is cancer-free for 3 years, sent us a letter that he was actually watching his son in the All-Star game playing baseball. And I will show you why we were able to build this completely selective drug. Again, the goal was it has to be selective for only FGFR2, and the drug which was in the market was not selective. It had severe side effects because it also inhibited these homologous kinases, which we need for health. So that's why he had to come off, and he came to us and took ours. And so what was the key? The key was, you probably will know, protein dynamics. What we found that the cancer target, away from the binding pocket, this P-loop had a different dynamics in the cancer protein relative to the healthy protein, okay? And again, the binding pocket was identical. So what our team at Relay was able is to design an inhibitor, down over there, which takes advantage of the differential dynamics. So this is the drug over here. And what you can see, it covalently binds to the cancer protein and not to the other because it's too far. So ours one is fully selective for the cancer protein, and the one which is on the market before was actually binding better the healthy one than the non-healthy one. So with this, we were able to completely remove the toxicity which were there before. So the second one I want to show you, just one slide, because it is ongoing stage 2 clinical trials in a huge patients populations for breast cancer. This kinase is a big, you see kinases, PSV kinase is actually the most frequently mutated kinase in all solid tumors. And as you know, solid tumors are very hard to treat. And this was a very interesting target because the mutations, so the only ones which cause which cause cancer if those residues are mutated. And these mutations are away from the active site. Again, the molecule which is on the market is non-selective. It binds the mutant, but also the wild type, and has severe side effects. Our team was able to, again, discover a novel binding pocket in this movie over here shown by finding a pocket over here. You will see the drug coming in a second, over here, which is only there on the mutant protein, which is the cancer-causing one, and in the wild type, it's closed. And by designing that to this specific pocket over here, we now have a 80% tumor reduction with this molecule in combination with the second drug. Okay, so full circle, I talked a lot about how we use protein dynamics to make small molecules and drugs. But my first passion was these enzymes, which do these amazing reaction of catalysis. So I want to come back the last few slides to show us very, very new work from my amazing team, and they will actually do some demonstrations outside afterwards, my grad students and postdocs, where we developed a new enzyme which treats celiac disease and gluten intolerance. It's not treating yet, so we're having it just in the lab. So that's very ongoing early work. As you know, about 10% of the population is gluten intolerant and can't eat all this good food, which we will find out there. And there is no cure and treatment. So I asked the question, or as a team, what if we could develop a simple oral enzyme which you take, like the lactase against lactose intolerant people? So what's the goal? Very simple. We need an enzyme which cleaves this gluten. So gluten makes that immunogenic 33 protein, this guy over here, right? And this triggers this immune response. So we need an enzyme which basically just cleaves it up. Why is it hard? Because it has to work in the stomach, and the stomach is very acidic. So it has to be an enzyme which survives our acidic pH. It has to cleave very fast, and it cannot be cleaved by our housekeeping protease, pepsin, okay? So sounds not too hard, right? But it is hard. So I figured out a new approach. So of course, companies have been making these enzymes by directed evolution. And by the way, directed evolution is that you take a protein, you mutate the heck out of it, you make 10,000 variants, and then you screen whether you actually get winners. But often you get stuck. And I thought we get stuck because everybody's using modern proteins. So we used the same trick that we go back, this time 3 billion years back, and calculate a protein which actually lived 3 billion years ago. And the details why I think that these ancestors are more evolvable, meaning that we can find much easier winners, I can talk to you after the talk, but it worked. So it's an evolutionary concept. We used this old enzyme, did this directed evolution, and found winners. Of course, the other thing you have to do, if you want to actually find winners, you have to select, you have to make a high throughput assay. And my clever grad students designed that we have actually our gluten here, a fluorescence guy, which is quenched by another one. And if you cleave it, the quencher goes away, and we actually just get bright fluorescence. We can do this assay in cell lysates and 96 well plates. The cool thing is all the red guys are better than wild type. So we find winners on every plate. And the other thing is these ancestor protein, you can make really high yields, so it's cheap to produce. Indeed, our proteases are very stable, and they're active at this low pH. Okay, so this time travel back 3 billion years ago, and then to find the ancestor, and then in the future, by doing directed evolution AI, allowed us to indeed make a highly, highly fast enzyme. So these are the 4 generations. These are the mutations which we found to make the protein in every round faster and faster and faster. So we got a 700-fold speed acceleration with only 4 rounds, which is unprecedented, and it's because we started with this ancestor protein. So here's my gluten bold. That's where we want to go. Get it? So I showed you all these experiments. I showed you we have these amazing instruments here at Scripps. This magnet I showed you costs $2.5 million. You know, we have cryo-M, which are even more expensive. So what if we actually wouldn't have to do experiments anymore and could use AI to predict how these proteins dance? So that is now the new future where we are going in the lab. And the big problem is, of course, we know we want to do it, right? I mean, AI actually works pretty well to predict now these static structures, which are sitting in the valley, but that's not where they're active down there. We got to see them how they move over the mountain. And the problem is that there's not enough data to train a model. You all probably have used ChatGPT. And the reason it works so well because we have tons of language and we have tons of pictures. So an AI model needs a lot of data. So every time you use your phone, these new data are being used to train more. We have almost no data on protein dynamics. So we compiled a benchmark of 134 proteins where we actually have dynamics data. We tried to train, there was not enough. But we could use these data to evaluate the model we actually did train. So then I got this crazy idea. So the question is, how can we get from 134 proteins to thousands? To actually 10,000. I realized there's one thing we have in a database. We have actually 10,000 proteins people have looked at NMR, and we have assignments. And what are assignments? Assignments mean that our protein, we know where in the spectrum they are. So the black dots are all the amino acids which we see in the spectrum, and all the rest, the red ones, are all the ones which are missing. It goes back to me moving back and forth. I had this idea, everything which is missing in the database is actually because it's dancing. Because I'm moving so fast back and forth that you can't see me. It was a bold call, but it worked. So it's similar to what I show here. Yeah? If an atom moves back and forth, it actually broadens out. So we now use this very simple, bold idea, had 10,000 proteins, trained a new AI model, and now the output is just a German sausage. And the sausage is, when it's thick, when it's fat, it moves, and when it's skinny, it doesn't move. You can now predict for any protein you want this little prediction here. It takes 30 seconds. All you have to put in is your protein, and out comes your sausage. So now we know already with this new model where a protein is dynamics. So again, experiments take months and are expensive. This prediction with our new models takes 30 seconds. Now we're on the age of, I can tell you anything, right? Remember I told you I will show you some data. So my last slide will be some data to show whether it's actually scientific Hollywood or whether what we are predicting is correct. And with that, remember I told you we had the 134 proteins. When we have data, we took it out. We never used it for training. Now we can ask, how does our prediction, which takes 30 seconds, compares with these experimental data? So remember, the sausage is where it's fat. Our model predicts it's dynamic. Where it's skinny, it predicts it's not dynamic. And then the color is where we actually have NMR data where it moves. Hello, the fat stuff is colored. So indeed, our model predicts where the protein is dancing. Not perfectly. We're far from perfect, but it's a huge breakthrough. For instance, KRAS, it's a huge cancer target. 30% of cancers have KRAS mutated. And this is a hotspot. and it's lighting up like a Christmas tree. What we see here also that our model predicts actually these motions in biological important regions, which are active sites, binding sites, and so on in enzymes. So, just to close today, I hope I have shown you how little we as scientists actually know. The cool thing is, all the young grad students, undergrads, kids, I'm very excited how much better we can do with the new tools we are developing. So we are just scratching the surface. I showed you that we can design an enzyme, but it's still not as good, not by far as good as what nature did. So there's a lot to do, and I hope that everybody is really excited to support the next generation, to support Scripps, which allows us to do this amazing work freely, crazy ideas. That's why I came here, because I'm a little crazy, and Jamie allows me to follow my crazy ideas. And just on the scientific part, I showed you that we are developing methods. So this is our new 800 megahertz spectrometer, which we just got half a year installed here at Scripps. Both experiments, but also how we use experimental data to finally use them to train new useful methods by AI to predict those things. And then on the right side, how we can use the knowledge of these dancing proteins to make better enzymes, for instance, the glutenase. And we are very hopeful that we actually will have a molecule at some point developed by a company which can treat gluten intolerance. And this big concept of making really medication which minimizes the side effects by this double drugging. The most important slide is this, of course, the dream team. You will meet them all out there. So that's my current lab here at Scripps. And of course, thanks to the real MoMA team. Very big thanks to HMI and the Scripps Research Institute, which funds all that work over here, right? And finally, the work I presented was not done in a few years. So this is actually my Kern gang from when I started my own lab to today. You can see lots of colors, lots of happiness. Science is amazing. And I want to particularly thank my father, Gerd Hübner. He's actually the one who taught me how to measure kinetics. The best lecturer ever in kinetics and enzymology. And we had actually a couple of papers together. And then Nadia and Gunther, who are in the audience, who were instrumental on this Aurora kinase to figure out this multi-step binding of drugs and how to make selective drugs. And of course, I thank you for listening.
So, thanks, Doro. Thanks so much. So we have time for some questions. So just don't be shy. Stick your hand up, and one of the mic runners will find you. So I was really interested in. I mean, everybody's fired up about AI, so that was a really nice thing at the end, and so. But the first part of your talk was all about how small molecules bind to proteins and change the dynamics. And so can you figure out, that would be the next step for AIs. How do you think about coupling the dynamics with the small molecule binding and using it?
Fantastic question. This, by the way, is what a lot of companies try to do. The issue right now is that we don't have, again, enough negative data. So everybody knows the positive data, the drugs which are working. I have been really advertising for a long time that we should also have all the molecules which actually failed. Not even for the binding. I think the harder thing is actually in clinical trials, right? But even for the binding, I think what you need is a big enough data set, but it's, again, you're training for a certain kinase, right? So it is happening. I think I'm very hopeful. The key is that scientists work together and also companies work together to actually share data. I showed you in the back. The key is good data.
Yep.
I haven't even talked about it because the quality of the data is really bad. And the quality of affinity data for drugs is very lousy. Mm-hmm. So if you have lousy data going in, lousy data come out, right? So I think it's
Bad
a quality.
sausage.
It's a quality we need and the amount of data to share to do it. But it's going to happen.
Great. So do we have, wait, there's one down here. Yep, just go for it.
Test. Okay. So my question is, when you're talking about the proteins dancing, okay, and I'm non-scientific, so when you're talking about them dancing, is there a foundational dance that, let's say you have protein A, B, C, and D. Is there a foundation that they all work on, or how many different steps, and maybe we don't know that until A, how many different steps are in that, what you call dance, and is it, how do I put it, is there a time duration that allows it to, the drug to get in there at certain steps of that dance to be able to go? Does it happen one time in every X amount, or does it open 3 or 4 times?
Yeah. So very good question. So the time is actually, I'm glad about this question, right? The timing, I didn't talk about it, how fast these things, I mean, I gave some numbers. Some proteins move, there are movements in picoseconds, nanoseconds, milliseconds, seconds, and so on, right? The important movements for the biological function is actually dialed in by the biological function. So for instance, we work on a protein which is in the nervous system. So the motions have to be super fast because your reactions are milliseconds. We're working on another protein which controls your circadian rhythm, 24 hours. Guess how slow the movement is? Several hours. Because, right? So it's controlling a very slow biological process, right? So you have the entire time scale, and I think, and so the key thing for drugs, right, where this dance, where I said the pocket is open and so on, but then this moving over the drug. If you paid attention, which I did not actually emphasize, getting out of this took actually 30 minutes. And that is the key to design in, that the movement in the drug-bound state is very slow. And the reason is because if it's very slow, the drug stays on this protein way longer. And the residence times of your drug on your target is absolutely essential. So we actually want to dial in a very slow, stable complex where we can't get out.
We have over here, yes?
Yeah, in the middle. In the middle here.
Oh yeah. I mean, I feel like I'm in Hollywood, like I'm like this, I can't even see you guys.
That was really fascinating and fantastic talk, thank you. I wanted to ask a question about the modeling at the end. When you identify those regions that are more prone to be dynamic, do you get any sense from the modeling of what are the features of those regions, the amino acids that are there, like the properties of those regions that are then being predicted?
Yes. So very good question. So one question here is the type of amino acids, whether this actually contributes to it. And we found some trends, right? For the histidines, and so they are moving more, but also because they can be proteinated, non-proteinated. But what is really the importance there is, and I didn't want to go into technical details, we are actually predicting correlated motions. That's why the dance is a good analogy. You have fast local movements where a little atom just goes crazy. But all the movements which are actually important for biology and drug binding, they are dancing together like a domino principle. So the atoms, when you push one, the next one goes, the next one goes. And so it's this correlation, the correlated movements together that the whole body goes. That is actually what we're predicting. And that's what we cannot actually do right now if you just do molecular dynamic simulation. So it's really a very correlated dance together, which we're predicting.
Great, thanks. We have one down in the front. Did we have one here? Hannah? No? Another question? So, I had another question. So, you gave this really great example about Gleevec.
Yes.
It was a wonder drug, yet it had some shortcomings.
Yes.
So, there's tons of drugs on the market. So, for how many of them is this soft underbelly of, suboptimality, right? Is this a rare case, or is this the usual case? And, are you going to be able to improve all drugs, or do you just have to find the ones that are set up for this approach?
I think you can improve all the drugs. And this double drugging approach, I'm really advertising for that, right? Yep. So Gleevec is actually a great example. Because of time constraint, I took it out. So Gleevec is very selective, but we have this problem of resistance. So the first clinical trial of double drugging is actually with Gleevec and another radiculocyte inhibitor from Novartis. And there is, the patient data is no resistance anymore. We looked at that and found out that the combination they use actually is almost a tug of war. They're not working together, right? So I think by understanding this biophysics a little better, and we found now one which actually is synergistic, which works together, right? Very quickly, right? So I do think that this to me is one of the best approaches to improve the drugs by adding two together and to avoid this resistance. Yeah, Gleevec is a great example, right? It works well.
You know, the other thing that really struck me in your talk, and it was a theme in the two parts, was you used what you called ancestral proteins.
Yes.
So you kind of trace back. So these proteins don't currently exist.
No.
But you can kind of infer where they came from.
Yes.
And so what is the, what's the special sauce? What are the properties of these ancestral proteins that allow you to develop present day properties?
It goes back to evolution is really, really cool. We are all a product of evolution. The reason we are sitting here, because we are the fittest. Everybody who's not as fit as us is already extinct. All right, so good news, congratulations, we're all the fittest, okay? But I bring this up because this was my theory why I said the ancestors are more vulnerable. It's not that all the ancestral proteins 3 billion years ago were fitter. We are calculating back that sequence from all of us, from the fittest. So if we combine all our fittestness, because everything else got extinct, we are actually calculating back then the combination of the fittest guy. So that's number one.
So T-Rex isn't in there.
No, no. He wasn't as fit as us. He's gone, right? Does that make sense? And that's why we don't violate evolution. So 3 billion years from now, we're still evolving. If something happens catastrophically, some of us will actually live, right? But only the fittest ones. So it's the way we calculate these ancestors, right? And so we are multiplying good properties, basically, right? The other thing what we're finding, because we do that, is that we can make a lot of them. They're very soluble, right? They're just very robust, yeah.
Okay, any other questions from the audience? This is your last chance. But there's one more down here. Great.
I was just going to ask a side note real quick about the dance. What do you think about, because you're into athletics, and I haven't had a chance to talk to you since Jason Tatum came back to your favorite team. Do you think that's going to mess up the synergy, like the protein dance, because they're doing so well right now and they're going to insert him? On the record, give me your opinion on that. You're going to be held to this now.
He is doing just fine, Tatum. I have watched the last games, and he is a little tentative. So I think it's going he's doing his right thing.
Okay. Sorry, that one, for me, that was just, I don't know.
Basketball and the Celtics.
Okay, okay, got it. All right.
Basketball and the Celtics.
Okay, that's all right.
I'm a basketball player.
It's good you're multifaceted. I appreciate it. Okay, well, I didn't mention this at the beginning, but I've been trying to recruit Doro here for well over like 10 to 15 years.
More than 15 years.
Yeah, and so I was finally successful, and I'm glad you came, and I was right all along that you're a spectacular scientist, and it was a really engaging lecture. So for the next lecture, we have a special treat. Pete Schultz is going to come and talk about his vision for science and for the Institute, and that is on, it's April 18th, I believe. April 18th, it's Pete. It's April 22nd, thank you. So I'd be alone. But hopefully you can come. If you can't come here in person, please join us in the virtual audience. For those of you who are here, we have a little reception out. And on April 22nd, you will find me in the front row.